In the lab this week, you are going to be given 3 species of Staphylococcus and 3 species of Streptococcus samples, but the identity of them will not be known to you. And so what you would do in this lab is to use those tests that I described in the lesson to identify what species each of the samples is. So in this lab, each table would start out with a set of broth cultures, and you'd have 6 cultures, and they're only labeled as A, B, C, D, E, and F. And so your job in this lab would be to identify each of those cultures as either Staph aureus, Staph saprophyticus, Staph epidermidis, Strep pyogenes, Strep agalactiae, or E. faecalis. You'll have one of each, but you won't know which is which. So first, we want to start by separating the staph from the strep. And there's an easy test to do that before we start the rest of the tests. And that is the Catalase test with the hydrogen peroxide. So staph are catalase positive. They reduce hydrogen peroxide to water and oxygen, and they produce bubbles. Strep is catalase negative, so when it's mixed with hydrogen peroxide, you would see no bubbles. So you're going to test each unknown for the presence of the catalase enzyme. So place a small droplet of each bacteria onto an empty Petri dish, and then to each droplet add a drop of hydrogen peroxide. So if bubbles occur, you know that that strain is staph. If there's no bubbles, you know it's strep. So now you can divide your 6 samples into staph and strep. So after performing that Catalase test on all your 6 samples, you would have determined that B, C, and F are Staphylococcus, because you would have seen bubbles for those, and you would have determined that A, D, and E are Streptococcus. So now you're able to divide your samples as either staph or strep, and then the following tests you will do depending on whether you're trying to distinguish the staph species or the strep species. So the first test is the coagulase test, which you would perform on only those 3 strains that you determined to be staph. So you would thoroughly label your 3 rabbit plasma tubes that you would be given with the name, date, and bacteria letter. And then using a fresh disposable pipette for each tube, you gently squeeze 1 to 2 drops of the appropriate bacteria into the tube containing the rabbit plasma. The second test is a test you would perform only on the 3 species that you determined to be strep, and it's called the bile escalin test. And you start with these 3 tubes of solidified agar called bile escalin in these tubes, and you would thoroughly label your 3 bile escalin slants with the name, the date, and the bacteria letter that you're inoculating with them. And then use a fresh swab for each slant. Take some of your culture, and then in the slant with the corresponding letter that you've labeled, you're going to gently zigzag a streak of the bacteria up each agar slant, starting at the bottom and gently zigzagging up towards the top. The third test is the novobiosin and bacitracin sensitivity test. So the antibiotic sensitivity. So you're going to use TSA plates and thoroughly label them with the name, the date, and the bacteria letter that you're testing. And then using a fresh sterile swab, you're going to streak along onto the plate. So you can even use a pipette and get some bacteria and just put that onto the plate first and then spread it around with a sterile swab. And making sure to use sterile technique, really spread the liquid around on the TSA plate until it's totally covered. And then you're going to let that dry for about 2 minutes. After that's dry, you're going to use the incinerator to sterilize a pair of tweezers. And then you let that cool for about 30 seconds. And then you use those incinerator sterilized tweezers to place the disc of the antibiotic onto the plate. So you use it to take out a disc of the antibiotic and put it on the plate. You would use a bacitracin disc in the center of the 3 strains that you determined to be strep, and you use a novobiosin disc in the center of the 3 strains that you determined to be staph. And then the 4th test is the MSA plate test, or mannitol salt agar plate. So you divide each plate into 3 sections, and one plate will be, you'll streak the 3 staph strains, and the other plate you'll streak with the 3 strep strains. So you're going to thoroughly label each plate with the name and date, and then on each section, put the label with the bacteria letter. And then using a fresh, sterile swab for each bacteria, you're going to take some bacteria and swab it carefully onto the section labeled with that letter. And do that for each sample. So all 3 staph strains on one plate and all 3 strep strains on the other plate. And as you do this, you have to be very careful not to cross-contaminate. So that means don't let the different bacteria touch each other. So don't cross the line that you've drawn on the plate. And then you're all finished. You can incubate everything at 37 degrees in the incubator for 24 to 48 hours. And really important to clean all benches with bleach, because some of these, remember, are really pathogenic bacteria.